Hello there and welcome. I'm Dr. Hakim and today's topic is about Staphylococcus aureus. So, first of all, we should have an understanding of the bacterial nomenclature. Staph means a cluster and cocci means round or spherical, which means in total that this bacteria has a spherical shape. It's a cluster of spheres. Here's an overview of this bacteria under a microscopic picture after gram stain. Now, let's dive deeper into the characteristics of this bacteria. It's a gram-positive because it retains the crystal violet after its coloration, and that's due to the peptidoglycan layer that it contains, which makes her appear like a bunch of grapes. It's a non-motile bacteria, which means that it has no flagella that enables her to move in different areas. It's a facultative anaerobes, which means that this bacteria can survive in both the oxygen environments and non-oxygen environments types. All Staphylococcus bacteria has an enzyme called catalase, which means that if we put these colonies into hydrogen peroxide, it will be transformed into water plus oxygen, and a bubbles will result also. Let's talk more about the medical microbiology part. Where it's found. This bacteria is simply found in the human body. The Staphylococcus aureus is a part of the natural skin flora. It can be found in the armpits, in the ears, in the pharynx, in the nares, and in the perineum, which leads to the higher risk of infection of the female urogenital tract. Compared to Staphylococcus epidermidis and Staphylococcus saprophyticus, who have a common characteristic of containing urease enzyme, the Staphylococcus aureus is differentiated from them by being the only species that has the coagulase enzyme that can convert the fibrinogen substrate into fibrin with the formation of clumping of the solution. The culture of Staphylococcus aureus in a petri dish that contains the mannitol salt agar leads to the appearance of the golden yellow colonies as a result of the fermentation of its nutriments. This color is the origin of its denomination Aureus. Here's a picture of it. If we come to the pathophysiology and mechanisms of infection, we will see that, in fact, this bacteria has a couple ways to cause damage in the human body. The first one is by a biofilm. The exopolysaccharide layer protects the bacteria against the immune cells, which enables her to escape from the immune system of the body. It's usually a catheter-associated infection or a urinary catheter insertion. Also, it can be a result of a surgical procedure. So, as I said before, we have this bacteria already existing on our skin. It's a part of a natural flora. When we try to put the catheter inside the vessel, a few number of this bacteria will be attached with this device to get inside the vessel of the blood. And then we will have the next result in the picture. The device has a few number of bacteria attached with it after an initial attachment, and then we will have a multiplication of this bacteria and accumulation. It will undergo through maturation and then detachment and dispersal into the blood circulation. The second mechanism besides the biofilm is the exotoxins, such as toxic shock syndrome type 1. This toxin, after being released into the bloodstream, it will act as a superantigen and bind on to the receptors of the antigen-presenting cells. And then it will form a complex that will hyperstimulate the T cells to a massive release of cytokines such as interleukin 1 and 2, TNF-alpha, and interferon gamma. This massive release will lead to a massive inflammatory reaction on skin, translated into rash, a general vasodilatation that leads to a hypotension, which means a low blood pressure. Also, a feedback interaction of inflammatory substances with a hypothalamus reaction will lead to increasing the temperature of the body, which means fever. The toxin is leukocidin. This toxin, after being released, it can induce spores on leukocytes, which leads to their necrosis. And massive liberation of its content may cause inflammation damage to lungs tissue, leading to necrotizing pneumonia. Another type of toxin is exfoliative toxin, which reacts on keratinocytes of the skin, specifically on the desmoglein type 1, which is a protein link between cells. The damage of this protein will give a result of a red rash, a blister, which gives us the Nikolsky sign, positive, which is a presence of poor oral hygiene associated bullous lesions and ulcerations. Nikolsky sign is an epithelial cells are separated from the basement membrane, allowing an assessment of the fragility of epithelial attachment mechanisms, acantholysis. Here's a picture of that Nikolsky sign. It's called staphylococcal scalded skin syndrome, a blisters of the oral mucosa. The next dangerous toxin is hemolysin beta, which has the ability to destruct the red blood cells, which means if we put the colonies of Staphylococcus aureus in a blood agar plate in a petri dish, we will see the next picture, where we will find an empty spot of blood. That's due to the ability of this toxin to destruct the red blood cells. Now we see a blood agar showing golden yellow colonies with beta hemolysin. The last exotoxin is the enterotoxin, which reacts on the enterocytes of the gastrointestinal tract to induce a gastroenteritis. This reaction will result in the destruction of these cells, and sodium and water will leak from these cells into the gastrointestine, which will lead finally to the symptom of diarrhea. Now let's discuss about an interesting mechanism, which is Staphylococcus saprophyticus and urinary tract infection. So let's say you have a patient and he has a urinary retention. The right thing to do is to put a urinary catheter, but with this move you have a high risk to introduce Staphylococcus saprophyticus into his bladder, as we spoke before. So you will introduce some of these bacteria into his bladder, and then a biofilm of this bacteria will be formed. And then the interesting about this bacteria is that it has a urease enzyme, as we said before. This enzyme has the ability to convert urea that is contained in the bladder into ammonia plus CO2. But the problem is the ammonia will be modifying the pH of this milieu, and the higher pH is, the bacterial growth will be much more, and the result is cystitis. Here's an overview of a bladder and a bacteria getting up through the urinary tract to the bladder. Also, the ammonia will combine with magnesium and sulfate, and that's going to lead to the formation of struvite crystals. This struvite crystals will lead to the bladder obstruction, and the infection goes up into the kidneys. Now, let's see different forms of diseases caused by Staphylococcus aureus. So, after the skin infection and through contiguity, many tissues and structures can be affected, included but not limited to, for example, the hair follicle. Its infection is called furuncle. A bunch of furuncles are called carbuncles. If we move to a deeper part of the skin, which is called epidermis, its infection is called impetigo. If we move deeper to a next part of the skin, which is called the dermis, its infection is called cellulitis. If the infection goes deeper into the muscle part, the infection is called pyomyositis. Another type of the infection is called abscess. If the infection goes deeper into the bones and articulations, it is called osteomyelitis and septic arthritis. There are other dangerous types of infections caused by hematogenous spread, or what we call septicemia, which means through the bloodstream, the bacteria moves to the meninges and can cause meningitis. Or if it moves to the lungs, it will cause pneumonia. The other case is the infection of the heart, which is called endocarditis. How do we treat these infections? The treatment is with antibiotics. First of all, we have the medicine-sensitive Staphylococcus aureus. Even though it has the beta-lactamase enzyme positive, but it is very sensitive to antibiotics such as oxacillin and nafcillin. But after that, these bacteria have developed a resistance against these antibiotics, and it became methicillin-resistant Staphylococcus aureus because it has the MECA gene that gives her the ability to develop a protein called PBP2 alpha. This protein can diminish the methicillin efficacy. So the methicillin efficacy is diminished by this protein. And now we distinguish two types of MRSA. The hospital-acquired MRSA, which is treated using vancomycin, and the community-acquired MRSA, which is usually treated by using doxycycline, clindamycin, or Bactrim. The bad news is these bacteria developed another resistance against these antibiotics, and we called it vancomycin resistant Staphylococcus aureus because of the VAN-A gene that can alter the peptidoglycan cell wall. And now the molecule which we use to treat this type of bacteria is linezolid. Let's finish with the last part of prophylaxis. So, one of the solutions is removing the temporary medical devices such as catheters if possible, hygienic conditions for medical procedures, and appropriate use of antimicrobial drugs. Proper environmental control methods, including the regular monitoring of air, water, and surfaces. Stringent cleaning and disinfection of equipment and environments. In clinical environments, isolating patients when appropriate. Thank you for your valuable attention. If you'd like to enjoy more of our lectures, you can visit quadia.com. See you next time.